know when, maybe tomorrow. It won't be super quick. All right, so let's take a quick look here at chapter 1, and I'm going to just grab my book if you guys can do it. So, as I mentioned, the chapter 1 is all about the themes of biology and what unifies the study of all biology. And chapter 1 in every textbook is the same. It is basically, here are all the things you could possibly learn about biology if you studied this whole book. And what you guys need to realize is that we're not covering everything. Okay, we're going to cover mostly concentrating on the cell and a little bit on organisms toward the very end of the semester. So this first panel here is the first theme, which your book titles organization. And part of the organization of living things, because biology is an ology, so it's the study of bio or life. Part of that organization is to characterize what features must an organism have in order to be considered a living thing. Okay, and your book goes with these, I think there's 7 or 8 of them. 1, 2, 3, 4, 5, 6, 7. Okay, so let's just start with the first one here with order. And what they mean by order is how different parts of the cell or different parts of the organism perform different functions. So you could put for here, structure dictates function. Second characteristic that you must have to be a living thing, your textbook is titling it energy processing. You may have learned that in the past, in your past somewhere as metabolism or ability to make energy and how an organism utilizes energy. We will have 3 chapters to talk about energy processing. Again, focus primarily on how cells process energy, and remember in cells and in organisms, the type of energy we're going to be talking about is ATP. It's a form of chemical energy. In order to be considered a living thing, there must be growth and development. It's pretty standard. Growth, we have a seed here, right? Growth, it's getting taller, and the development, it's changing from one form to another. So this is growth and development because some organisms are like a bacteria, for example, or an amoeba. They don't develop or change very much, but they certainly do grow and get larger. I'm going to jump here to the one that's related to that, reproduction. In order to be considered a living thing, you must have some form of reproduction. This could be sexual reproduction, as in the case of animals and all plants, or it could be asexual reproduction in the case of protists and bacteria. These two, regulation and response to the environment, even if you're a single cell. So let's look at response to the environment first. We've got a picture of a Venus flytrap here, eating a grasshopper. That's a response to touch receptor, right, in the plant. As a complicated animal organism, you respond to the environment all the time. You change your that's the easiest example would be your pupils dilate, and when you are exposed to a darker room so that more light can come in, that would be counted as response to the environment. Even if you're a single cell, you have the ability, even single cells have the ability to usually move, for example, toward food, away from a toxin, toward prey, away from a predator. That would count as response to the environment. Regulation, we have this little bunny here with the giant ears, right? He's regulating his heat. He's doing thermoregulation there by dissipating heat through his ears because he doesn't sweat on the rest of his body. Again, we're going to be thinking about regulation in terms of inside a cell as well as inside a whole organism. Another word for regulation in terms of the organism or the cell compared to the environment, we would say homeostasis. And this last one here, evolutionary adaptation. This is the overarching theme of biology. And you can see here there's a seahorse that is perfectly blended into his environment. that looks like coral, even though he is not coral, right? In order to be considered a living thing, at least in biology, you must meet all of these characteristics. So for example, a virus, which infects living things, it does not meet all of these characteristics because it does not process its own energy, and it doesn't technically reproduce, it multiplies. So it does not meet all the characteristics of a living thing, so it would not be a living thing. Now, the second part of this theme of organization is not just what are the characteristics that you need to be a living thing, but how do we group living things and study living things? And this I put on as letter B here, as structural organization, and we're starting with the whole globe, the biosphere, and we're looking at smaller and smaller units of organization inside of living things. Okay, so the whole biosphere and ecosystems, how living things interact with the physical environment, communities, different types of living things together in the same environment. So we have animals and plants here in this picture. A population, which is defined as a group of organisms that are the same species living in the same place at the same time. That's an important definition because a population, we're going to see, is the smallest unit of evolution. So that's going to come back to us at the end of the semester. Here's a single one of those, look like delphinium plants here. We would call that an organism. And then the separate parts of that organism, we refer to organs, and we can break down those organs into specialized tissues that have singular or a very small subset of functions. And then those, of course, could be broken down into cells. It's a picture of a mesophyll cell here full of chloroplasts. The organelles inside the cells, picture of a chloroplast, and the molecules that make up those organelles. I believe this is actually a picture of chlorophyll, this molecule. In Bio 1, we are going to spend almost all of our time right here. Molecules, organelles, cells, right? When you go to anatomy and physiology, you're going to spend a lot of time talking about tissues and organs and organ systems and the whole organism, right? And if you continue on and study environmental science or ecology, clearly you're going to spend more of your time here with ecosystems and biospheres. You study genetics, populations, and evolution here. I included this picture of the cell because I wanted to be sure to say to you that the cell is the smallest unit of life. So a single cell is the smallest entity that can have all of these characteristics. I liked this picture from your book. It is figure 1.4 because it includes the two major types of cells. This is called a prokaryotic cell, that's a bacterial cell, and this is a eukaryotic cell. That would include plants, animals, fungus, and protists. Now, the second theme is genetic information. And this is a complicated theme in that it also has two parts, right? The first part is how the genetic information of your DNA is converted or changed into proteins, how those proteins make up structures that give you capacity to do more than just an individual cell. So the example your book uses is a picture of a human with an eye. We're looking particularly at the cells in the lens. And then we're looking at the proteins that are actually made inside the lens that give it the ability to capture light. Okay. That protein is called crystalline, and what they're showing you here is the amino acids that make it up, and this would be the DNA that codes for that protein, and this would be a piece of DNA inside one of your chromosomes. I don't know which one, but one of them. This is one part of the theme of genetic information, and we have 2 chapters at least to discuss this. If we count studying genetics, I would say maybe 4 or 5 chapters studying about this theme, how a piece of DNA, how a code of bases is changed both through these processes of transcription and translation into protein, and how that protein functions in the organism to give us our particular characteristics. In this case, the ability to see. The second part of the genetic information theme is how this genetic information is transmitted through multiple generations. And so we have this picture here of how transmission through reproduction. So we have an egg cell. This would be obviously a human. We have an egg cell and a sperm cell, and we have a fertilized egg that's going to form a zygote, turn into an embryo, and then we get this adorable little baby here who has characteristics from both of his parents. I don't know, looks like a boy, maybe boy or girl, it's like a boy. Both of his parents, and think about the lenses of his eyes, he would have the variations that his parents had in their eyes because he would have inherited this genetic code from them to make this particular crystalline gene. Okay, so the theme of genetic information has these two parts, the transmitting of the information and then passing that information on to our offspring. The third theme, energy and matter. This is a very busy picture. It was the only one that was there, so I tossed it in. Basically, we have 3 chapters to discuss this. We're going to focus on energy flow inside cells. And again, we're going to focus on how plant cells can change light energy into chemical energy, and then how all of the animals and fungus and most of the bacteria use that chemical energy from plants to do their cellular work. I don't want to spend hours and hours on that. Like I mentioned, we have 3 chapters later for that. Theme 4 is your book very broadly calls interactions, and it also has 2 components, interactions within an organism. We would call these cellular interactions or molecular interactions. And in this case, they're showing you a feedback loop of how much glucose there is in your blood, how that affects how much insulin is produced, right? So this is all chemical. This is all chemical interactions of compounds being transmitted from one cell to another and then causing an effect that is going to affect the whole organism. Interactions within the whole system. And in this picture, your book, they throw in the whole ecosystem. So we've got plants and animals interacting and decomposition going on and gas production and the input of sunlight to produce the energy. So these interactions can become quite complex. And again, we're going to focus on mostly chemical interactions in Bio 1 that are happening inside a single cell. The 5th theme is evolution, and that theme I already mentioned, I think, to you is the overarching theme of the whole textbook. It's going to be mentioned in every single chapter. And these 2 parts here, these 2 portions, just to help keep it simple, one of them is classification, because we have millions of living things, and as humans, we like to put them in groups, and we like to put them in groups that share similar characteristics. So this particular slide here is showing you the current classification system, and that classification system includes 3 domains, and these domains are based on cell type. So domain bacteria and archaea, they are prokaryotic cells, simple cells without a nucleus. The domain eukarya, those are cells that contain a nucleus, and they comprise 4 kingdoms. These you're probably familiar with, right? Plants, fungus, animals, and the protists. I think I might hop out here and just, there's something that isn't included in the book that I forgot to mention here, in that so the largest group is domain, right? And we just mentioned that there are 3 of those domains, right? So we have 2 that are prokaryotic. So we have bacteria and archaea. H. My pen isn't working very well, sorry. Right, both of these domains have kingdoms underneath them that they didn't show you in that picture. In bacteria, there are 5 kingdoms, and they're based on different characteristics of those bacteria. And in the domain Archaea, there are 3 kingdoms. The Archaea, you can think of them as like the extreme bacteria. They are bacteria that live in really high temperatures, really salty places, and bacteria with really strange metabolisms. Come to microbiology, learn all about it. And then that 3rd kingdom of Eukarya, right, your textbook listed out those 4 kingdoms that we mentioned, fungus, right, protists, plants and animals. Plants, animals, and protists. Now, the panel that's missing that actually is not in this particular chapter 1 in your book, I think it doesn't show up until chapter 26, but I would like to include it, is the smaller groups below kingdom. So some of you may have learned this before, right? So we have domain. And on that first handout that I sent you by accident, the one that was from an older edition of the book, right? So we have domain, which we just discussed. And then the next smallest group is kingdom. The next smallest group from that we refer to as a phylum. Now, those of you that have a little more experience in biology, if you took an upper level biology class in high school, you may know that current terminology is switching a little bit from phylum to this word clades or clades, because that represents evolutionary history among organisms, and phylum or division is kind of an old-fashioned word from when we spoke in Latin in the 17 and 1800s. The most inclusive group is domain, and that's based on cell type. Then we have kingdom, phylum, and then smaller than phylum is class. And then smaller than class, domain, kingdom, phylum, class, order, family. Sorry about that. Family, and then genus and species. Most of the time in Bio 1 and in a lot of your other courses here at Camden County, this is what we'll focus on, mostly the genus and species. It didn't produce it very well, but notice that the genus is capitalized and the species is lowercase. And usually in a textbook, you'll see them in italics, both of those words, or when you're writing them manually like I just did, you would underline each of them with a separate line. This is the smallest, least inclusive group, the species, right? This is the smallest group here, right? And in this class, we're going to define that species as a group of interbreeding organisms that produces fertile offspring. I am not going to ask you to classify any particular organism. Like, I wouldn't expect you to know the classification, for example, of a polar bear. But I do expect, and I can promise there will be questions on the first quiz or exam about which more of these groups are more or less inclusive. So for example, I might ask a question like, if 2 organisms belong to the same family, they also share what, right? And the answer would be they're in the same order, right? Not the same genus or species. So that would be like a multiple choice sort of analytical question that I would ask. Oh, completely stopped my sharing. All right, let me try that again. Where are my controls? Yes, bacteria, the kingdom bacteria has 5 kingdoms and Archaea has 3. Yes, that's correct. All right, so I'm going to go back to my PowerPoint here. And so this was the first part, the classification. The earliest classification scheme was constructed in the 1700s by a man named Carl von Linné, who was a botanist. And at that time, everything was classified first into living or non-living. And if you were living, you were plant or animal. Clearly, in the last 300, 400 years, we've gotten a lot more sophisticated. And as I mentioned, the domain is based on the cell type. So back to our picture here. Prokaryotic cells, these cells without a nucleus, they make up the domain bacteria and eukarya, bacteria and archaea, and eukarya is of all the cells that have a nucleus. Okay, so then the second part of that evolution theme has to do a little bit with this man, and this is a picture of the front piece of his very famous book on Origin of the Species that was published in 1859. And the important takeaway here is that his idea that was sort of coalesced and more focused is that things evolve according to the laws of nature, meaning the environment is what selects which organisms are best fit for a particular environment. Notice in this picture, he looks a rather young man. This is right after he returned from his trip to the Galapagos Islands, where he, of course, studied the finches and looked at different types of finches. We don't have all 13 species here, but all 13 different species that were specific to the 13 different Galapagos Islands. I do like to include this sort of on the first day because it shows you what the current diagrams look like as far as what we would call phylogeny diagrams or systematic phylogeny, where you can see that we have an ancestral organism here, and then whenever there is a new characteristic or feature, it makes a branch point, and depending on how much information is available, these branch points will be sometimes spaced closer together or further apart, and at the bottom you might see a timeline, right? This might be, this isn't super informative because we don't know if this is 10,000 years or 10 million years, right? But in the later chapters, particularly if you go on to Bio 2, you will see many more of these, and this distance is somewhat representative of a particular amount of time. But in this particular example, we don't see that. So I'm going to add another screen on there because I did want to point out that, let's go withdraw, the two main important points. Oh, that's kind of smaller, right? To evolution by natural selection. Right. First is that new forms or new species, they called them forms in the 1800s, can appear. And this means they can appear in the fossil record, or they might appear in modern time, depending on how small the organism is. And that these new species are the result of variations among offspring. And the environment, and we mean both the physical environment of the climate, the temperature, whether there's water or no water, whether it's tall trees, the environment, the full environment, as well as predators, right, and plants and other animals that live in this particular environment. The environment selects the most fit organisms, and those organisms are going to reproduce and leave the most offspring. Okay, so I wanted to be sure to put those 2 points in there because I didn't put them on the PowerPoints, but if you looked at the old, if you looked at the note outline, it would be there. Okay, now let's go back. I don't know why it does that, but let's go back. And the last section of this chapter, the last theme, I would say, is the scientific method or scientific inquiry. And your textbook, again, divides that into sort of 2 sets of things. Observations and then hypothesis testing is probably what most of you think of when you think of the scientific method, right? And so here we have an observation: a desk lamp doesn't work. Why doesn't it work? Hypothesis 1, right? A hypothesis, remember, is an educated guess or a proposed answer to the question that was posed. Notice that the observation isn't the question. The observation is a statement of fact of what has been observed. The question, right, is derived from that observation. Why doesn't the desk lamp work? How does this cell work? Where in the cell is energy produced, right? Those are the questions. And then the hypothesis would be a proposed answer to that question. So this is the first hypothesis. Then you do an experiment, right? You test, you get some results. And if your results don't support your hypothesis, we go back to the question and we construct a second hypothesis. In science, so this is an oversimplified example here with something that we're all familiar with, a flashlight, a desk lamp. In science, a scientist may make 6 different hypotheses and test all of them before they have a better understanding of maybe clarifying or modifying the question that they're really asking. So this picture, your textbook puts forward as a more modern model of the scientific method.
Sorry
Right?
to interrupt.
So
you can't see the PowerPoint.
Oh, sorry.
Thank you.
Let me see if I can fix that. I lost my controls. Here we go. So the PowerPoints are already posted in Module 1, so you can look at them like the same time I'm looking at them with you, or you can look at them later. So this one was the one you couldn't see, right? So this is the simple scientific method here, where, like I said, we're using something that's very familiar to all of us, right? The observation of a desk lamp not working. Why doesn't it work? In your textbook, there's a nice picture also of as far as observation and setting up questions for hypothesis testing. There's a picture of Jane Goodall studying the chimpanzees as well. So this is I don't want to say sterile, but a very simple, in some ways, idea of what the scientific inquiry is or scientific method. Observation, question, hypothesis, test, analysis, and then back, new hypothesis if you didn't have the right answer to the question, right? And as I mentioned, in real science, a scientist may come up with 3 or 4 or 5 or 6 different hypotheses before they figure out why a particular thing is working or not working. So this model has been proposed as a more modern model of the scientific method, because what they're trying to show you is that your hypothesis testing is constantly informed by and changes based on new discoveries, feedback from the by community, they mean scientific community. There's some pictures of some scientists here having a meeting, a journal club with each other, as well as predicting or discussing some of the benefits and outcomes that are going to affect our whole society that we live in. So the reason they include this is because they want you to understand that scientists aren't working in a vacuum. Their new hypotheses are constantly being informed by all of these forces, right? The big blue one here, blue-green one, goes through more what we would traditionally think of as hypothesis testing / the scientific method, right? Doing experiments, gathering data, analyzing results, interpreting results, revising your hypothesis. I notice there's lots of arrows in this picture because they're trying to remind you that it's a process. It's not a one and done for anybody pretty much ever. And then societal, but I would put this one first. Scientific community feedback involves usually what we refer to as peer review. So as a scientist, we would write a research paper or we would publish our findings, and then other scientists would try to repeat them, or they would compare them with their findings and give us feedback, and that also might inform another hypothesis to be formulated. And then, of course, the benefits overall to society as far as the results of the scientific method, new technologies, informing policy, which we're literally in the middle of right now with the pandemic, trying to solve problems and building knowledge. And you could say all of those things are occurring right now in this particular pandemic with COVID-19, right? And this is in some cases driving the scientific method more than maybe normally, maybe than if I'm studying fish, right? So how much feedback you get from your scientific community, that's pretty much always the same, but how much feedback and pressures you get as far as basically funding comes from society depends on what's going on in the world in a particular time. And that, I would say, ends chapter 1. So I'm going to stop the recording.